Hello again, and welcome to the second lecture on microbial ecology. In this lecture, I would like to tell you a little bit about my own research on the honeybee gut microbiota. So honeybees have been studied for many years, and they are very fascinating organisms in their own right. But in our lab, we also like to think that the honeybee is a good model for the study of a natural bacterial community. A couple of reasons for that. You may remember from my last lecture that I told you that most bacterial communities are very complex, contain many different bacterial species. But the honeybee gut microbiota is a bit of an exception. It's actually remarkably simple. And that's a big advantage for studying the community because it makes the data analysis much easier. We also know that the bacteria in the honeybee gut are not found in other places. At least we haven't found them anywhere else yet. And so we have good reason to believe that they really belong there and that these bacteria and the honeybee have lived together for millions of years and adapted to each other. Now, important is also that we can do experiments on honeybees. By now, we know how to make microbiota-free bees in the lab. And we also know how to culture the bacteria that you find in the honeybee gut. And so in that way, we can take these bacteria and put them back into the honeybee in any combination that we like, and then study what happens. And there are some interesting parallels of the honeybee gut microbiota to the mammalian gut microbiota. I already mentioned that the community is specialized, as is our own community, but also honeybees are social, as are most vertebrates. And we know that the bacteria contribute to the degradation of pollen in the honeybee, so they have a nutritional role, just as the gut bacteria in our own gut. Now, the honeybee gut microbiota is not only simple, it also tends to be very stable, at least if you characterize the community composition at the phylotype level. So, phylotypes are here defined as groups of bacteria that have at least 97% sequence identity to each other in the 16S RNA gene. This is a threshold that is quite commonly used in the field. And here I'm showing you some data from a study that we published last year. It's actually based on metagenomic data, but in this figure I'm showing you the community composition as it would look also with 16S profiling. And so what you can see here is we have 5 phylotypes that are present in each and every bee, and therefore we refer to them as the core microbiota. We also have a couple of other phylotypes that we see quite frequently. But overall, we don't see any big differences related to the colony that the bees came from, or the year they were sampled, or the age of the honeybee. Might be a bit of a tendency towards a more unstable community in old bees, but that's about it. And this has also been shown in other studies. So there's not a lot going on at this level. However, we also know that the bacteria that belong to the same phylotype actually show some very big differences in the genome level. And so I'm not going to go through the results of this study here, just going to mention some highlights. So we did a metagenomic study in which we analyzed the community composition in a lot of detail. And one of the things we found was that these phylotypes I just showed you actually correspond to several different bacterial species. We also find that each of these species in turn have many different strains, and these strains differ in gene content. And we've looked at how the strains distribute among bees within colonies, and we find that each individual honeybee only carry a minor fraction of the strains that are present in the colony. And so because of that, the end result is that we have large differences in the gut microbiota gene content among individual honeybees within colonies, even though they look the same when you look at the phylotype level. And some of the genes that are variable are genes related to carbohydrate metabolism and transport. So it's possible that the honeybees differ in their ability to break down different kinds of pollen. So now I would like to show you some results of a very recent study that we have done. And in this study, we've compared the gut microbiota of the eastern and western honeybee. So when we talk about honeybees, most people think about Apis mellifera, also known as the Western honeybee, because this is the honeybee that is most widely used all around the world for honey production and for pollination services. But actually there are several different species of honeybee, and the most closely related one to Apis mellifera is Apis cerana, also known as the Eastern honeybee. These two honeybee species have been estimated to have a last common ancestor from about 6 million years ago. So the divergence time is about the same as between human and chimpanzees, for example. And currently, as I said, you will find Apis mellifera almost all over the world, but the native range is actually a lot smaller than that. Apis cerana, on the other hand, is native to Asia. And so these two honeybee species differ in their current geographic range. They also differ in several other ways. We know that Apis mellifera tend to make bigger colonies than Apis cerana. And although they look quite similar, Apis mellifera is also on average somewhat larger than Apis cerana. And some 16S profiling studies have already been done on these honeybees, which have shown that they have a very similar phylotype level composition. But given all the variation that we already found in Apis mellifera, we were wondering how similar these gut microbial communities really are. And so to study that, we formed a collaboration with a research lab in Japan, this Ryo Miyazaki's lab. And his postdoc, Shota, was kind enough to collect some samples for us. So Shota collected 40 individual bees from 4 different colonies, 2 from Apis mellifera and 2 from Apis cerana. And I have analyzed the metagenomes of these samples together with some of the samples we already had collected from Switzerland. And so firstly, here again, I'm showing you the phylotype level composition of all of these samples that we have included in the study. And so indeed, as was also shown in previous study, they do seem to have a very similar phylotype level composition. Here we have the 5 core phylotypes that we mentioned in blue and green colors. And as you can see, they take up a major fraction of the total bacterial community in each bee. We also have some phylotypes that seem to be specific to each honeybee, but they represent a minor fraction. Now, one of the things that we did in this study to make the analysis of diversity and composition within phylotypes a little bit easier was to culture bacteria from the gut of Apis cerana and then sequence their genomes. And in doing that, we actually discovered several new bacterial species. Here I'm showing you phylogenies for 2 of the core phylotypes that colonize both Apis mellifera and Apis cerana. And you can see here that the bacterial genomes form into different clusters. And the blue ones all contain isolates from Apis mellifera, whereas the red ones contain isolates from Apis cerana. And if we then look at how these bacterial species distribute across our metagenomic samples, you can see that there is a very clear host preference. So the blue species up here colonize Apis mellifera, and the red ones colonize Apis cerana. There's also been a few transfers if you look carefully in the bar plot here below, but overall, the honeybees are apparently able to maintain distinct bacterial communities. And in fact, we found this pattern for all the community members in the guts. And so one of the results of this study, a very important result, is that although they look similar at the phylotype level, the communities are actually highly distinct. Another result that came out of this study was that we found that the diversity of the gut microbiota is much higher in Apis mellifera when compared to Apis cerana. Now we've analyzed this in different ways. I'm just going to show you one result here. So what I've done here is that I have taken the genes predicted for each metagenome sample, and then I have clustered the sequences based on sequence identity. In this case, I used a threshold of 95% nucleotide identity. And so in this way, you can get an estimate for the number of non-redundant genes. And if we first look at the level of the individual bees, here's a box plot that shows how many non-redundant genes we find per bee. As you can see, we have almost twice as many genes for Apis mellifera compared to Apis cerana. We also find that the diversity within colonies is much larger for Apis mellifera. So here I'm showing you cumulative curves of the number of clusters. So the number of clusters you find in 1 bee, in 2, and 3, and 4, and so forth. And so the curve starts to flatten out here, as you can see for Apis cerana. This indicates that we wouldn't find that many new genes, most likely, if we were to collect more bees from these colonies. In contrast, the curve is still going up for Apis mellifera, which suggests that if we were to collect even more bees, we would find even more genes. And if we split the data according to the colony from which the samples came, the pattern still holds. So you can see even within a single colony, the diversity is higher for mellifera than for cerana. Now, this was a quite surprising result for us, and we still don't really know why it looks like this. But we have proposed a few possible explanations. So one thing we were wondering about is to what extent we as humans may have had an impact on the pattern. Because for Apis mellifera, this is a honeybee that has been transported around the world by humans for many hundreds of years. And maybe in addition to mixing the honeybees, we have also had a mixing of the gut microbiota. And that could, in turn, increase the diversity that we see within colonies. We're also wondering whether there could be an impact of diet. Currently, we don't know so much about the foraging range of these bees. There's been a few studies in Asia which indicate that they forage for more or less the same things, but it's possible that there are some differences. And if Apis cerana turns out to be more of a specialist, then maybe it doesn't need such a diverse microbiota. A third possibility is that this has something to do with the bacterial habitat size. So there is a very known, almost law in biology known as the species area relationship, which states that in bigger habitats and more complex habitats, you should find more species. And indeed, that has been shown to be the case for plants and animals many times. Whether it also holds for bacteria is still not really clear. But in our system, we do know that there is more bacteria in the gut for Apis mellifera compared to Apis cerana in each individual. And as stated, we also know that they make different size colonies. And there are differences both in their native range and in their current geographic distribution. Now, these two studies have raised a lot of new questions that we would like to look into in the future. We still don't know why individual honeybees carry different bacterial strains within the same colony. It's possible that this is to do with priority effects. In other words, the order in which newly emerged bees encounter bacterial strains in the colony. But it could also be that it has something to do with differences in diet or behavior. And we would also like to look into whether these genetic differences really have functional consequences. One difficulty with metagenomics is that we have many genes that are of unknown function. And it's also possible that some of these genes are actually doing the same thing, even though the sequences are different. So for that, we need to do some experiments. And finally, we are also wondering whether this difference in diversity that we see between Apis mellifera and cerana has any fitness consequences. Is it good for the honeybee to have a more diverse community? For example, by having more different genes to use for carbohydrate metabolism? We really don't know at this point. And with that, I will end this second lecture with a picture of the people working in my lab and also a link to our webpage. If you do want to know more, feel free to take a look at our webpage and also to contact me directly, and I will be happy to answer any of your questions.